Okay, so this map shows the mid-ocean ridge and Bangkok Basin and scattered hydrothermal vent or hydrothermal activities update up to 2020 in InterRidge global database of hydrothermal vent field. And so the yellow circle represent confirmed vent activities, and the black circle represent suspected, probably some chemical signature of vent activity below the water surface. So far, almost over 700 different vent sites were discovered by this definition, and if we see this line of mid-ocean ridge, you will see that it is marked with a different color, as shown here. Blue case ultra slow, and red super fast spreading rates. So we can see that the mid-ocean ridge system has a very different or various rate of spreading rate. So according to this rate, I think supposedly the geologic event would be different, and also probably the habitat features will be also different by this primary geologic environment. So today I will focus my study from the organism from Eastern Pacific Ridge. It includes EPR and PAR and also Galapagos Rift. Here you show that you see the high dense community of vent organisms like Liftia pachytula annelid and Tabunia Alvinella another annelid and Calyptogenia magnifica mollusk Bathymodiolus species that I will focus on my study this today, and the Bathograde crab like arthropod. And also, since the first discovery of this vent ecosystem in 1977 in Galapagos Rift area, continued ocean exploration and research extended our scope to the other ocean ridge system. Here I show the Central Indian ridge area and their vent communities. As similar as EPR, they also show very highly dense invertebrate communities. So this dense, abundant species flourishing is quite contrasting to the other ambient, just the plain deep sea floors. So since the past 30 years of population genetic studies have revealed a number of physical and biological processes that shape the geographical population structure and interpopulation connectivity and genetic diversity of deep sea hydrothermal vent species. For example, intrinsic factors such as taxon-specific larval development, larval duration, and maturity of larvae or adult and their behavior would result in some different types of behavior during their dispersal from one vent site to other vent site. And also the extrinsic or biotic factors like geomorphology of oceanic ridge and deep oceanic currents, as shown here one figures, and temporal stability of vents. In other words, spatial temporal presence or absence of vent activity along this area would influence to the vent organism. So here these figures was done by Lamiero. They characterize the development of hydrothermal vent, deep sea mussels. You know, they have a very different stage of duration and morphology, and also in relation to the symbiont. They rely on their nutrition energy. Regarding to the symbiont, they start to acquire their symbiont from the open ocean and environment, and then they grow their symbiont while they develop to adult. And as this yellow arrow indicates, the density and abundance of the symbiont increase within the specialized their gill cell bacteriocytes. And also, so we can see that the dispersal of this mussel would be influenced by current and the lava mediate dispersal ability, and also their due to their dependence on the symbiont. So the presence of symbiont would impact on their distribution pattern also, I think. And as another extrinsic factor, the water current regime would also influence on the dispersal of these larvae. When they flow along the water current, this direction of this deep water current would be influential factor for their dispersal. And also within our ridges, in a ridge valley system, the vertical warm flow or flow of direction of water current also would impact on their moving directions. So keeping in mind these environmental or biotic factors, the most intensively studied area in this mid-ocean system is Eastern Pacific area, where people studied the comparative the distribution pattern from Lithia, Tavernia, Oasis, Alvinella, and Brachypolino is a commensal of deep sea mussels and including mussel. So under this common geographical environmental settings, the biologists compared the genetic differentiation pattern from north to south. So as you see here, according to the taxon species specific differentiation pattern, like here mussel, they show slow gradual climb change along this latitudinal change, and then above they have a stop sign here, like you know, here it is the starting point of the Pacific Antarctic area, and other organisms also show differential kind of boundary of genetic differentiation. And so we can catch that not only the environmental factors, but also the organism-specific regions or factors would impact on their differentiation. You know, and also one common thing we can find with this previous result was that these organisms share some boundary, like equator and also eastern microplate area where begins new ridge system, Pacific Antarctic system. And also another point I can tell is that these almost all organism has very low genetic diversity within their segments. So that's the one, a few representative things we found from the past 30 years. Here I show the Eastern Pacific ridge system in more close. As you see here, you see the bottom bathymetric map of the Eastern microplate has high elevation than other ambient sea floor. And also it is a different shape of a linear ridge system. It shows round shape of elevation in that area. East Island here. And each ridge segments or ridge area has different spreading rate. Here 85 to 150 millimeter per year spreading rate. However, the Pacific Antarctic region has over 140 millimeter per year. So along with this bathymetric geological topology, also the ocean circulation study shows that this bottom bathymetry influence on the direction of water flows. Like here, Fujio et al. They reported around the Eastern microplate there is a wide anti-counterclockwise direction. That is right angular to the EPR region. So it would be, this direction would be impeding direction to the dispersal of bent organism navi. And also here another third evidence suggests that there is directional bias of helium element that it has original vent. So according to Lupton, he showed this wide area helium distribution map. We can see the westernward biased flow directions from here, the impact of that. So keeping in mind this type of the dispersal barrier or physical obstacles to the movement of deep ocean bent organisms, the one biological, in the field of biology, the method that we can detect the influence of such a physical barrier to the biological organism is kind of a population genetic model. It is called isolation with migration model. It incorporated the long traditional idea of Charles Darwin's evolution that understand the history of biology as a speciation event from the ancestor to the splitting to descending population here, N1 is called N1 and 2. And so after splitting, these two descending current population might have a certain rate of migration to bidirectionally. So this population genetic model includes 6 parameters for this major event: effective population size of city population and splitting time and migration rate. So these 6 parameters can encompass very diverse or various history of organisms on the earth. So I would here show some idea how biologists, particularly evolutionary biologists, envision the event or history of life. So here, as one population or one species goes from past to present direction, this the splitting of this load indicates speciation event at certain time. Within this population or species, they might inherit their genealogy. Gene might be inherited from ancestor to their descendant. And we also have a molecular change of DNA, like mutation event. At the end, it will end up with this type of variation pattern between two descending populations. However, this event is not such a single event. It could be different history like this. Here, the dashed line indicates migration. So a mutation event in here, it will be shared by other neighboring or sister population. And at the end, it will be ended with this pattern. So this left side and right side would be similar in terms of variation. So it would be very diverse, huge possible scenario on the evolution of the species or population in with respect to this genealogy and mutation variations. So IM model intended to incorporate this scenario under the statistical probabilistic model. So they do like the Bayesian approach for this population divergence using this 6-parameter model. And they use Markov chain Monte Carlo simulation to solve a probabilistic equation to get the estimate of 6 parameters. So this model, genetic model, is based on coalescence, and also the result as shown here, left side, it is curve of certain parameter here, time splitting time, and this the value of time will have probabilistic value curve. So, and this in 2001, Nielsen and Wakely designed this model from the two population comparison with one single locus. At the time, it was mostly mitochondrial DNA because it was available easier at the time. And since then, Professor Jody Hay in Temple University, he continuously developed this approach, developing program and adding more parameters or implementation, like here likelihood ratio test between the migration rate, or it expanded to 3 population comparison. So it becomes more realistic to include the real situation. And recently, Eugene Chung and Jody Hay, Eugene Chung is now here with us, and she developed another extension. So thanks to this final, this recent development, we could now use more than around 1,000 different genetic loci DNA data. So such a huge amount of genetic data is now become more doable, possible, due to the technology, DNA sequencing technology development, next generation sequence. So we could get such a huge amount of genetic data and can calculate very complex model parameters. These parameters deals with the history of species in nature. So come back to the deep sea. I will focus this application with these 3 species, Alvinella pompejana in Eastern Pacific, and Bathymodiolus, 2 Bathymodiolus species in EPR and Central Indian Lychee. And also interestingly, I attempted to use the IM method to the study of evolution of environmental or symbiotic bacteria. As far as I know, this is maybe the very early application of this method to bacteria. So first, Alvinella is a very charismatic species in hydrothermal vent because they live on chimney. They live on hydrothermal chimney, very hot temperature, and they have a characteristic pioneering species. When you bent come out, they are only settle for their new chimney, and they have a large lexicotrophic larvae, and also, they have a characteristic of larval development in cold water. When they expose to cold water temperature, their development arrest. So such arrest extend the larval stages. So finally, these larvae could disperse very long distance. So keeping this in mind and keeping this geologic setting around the PR, they have this ridge system has two known boundary of ridge segment where genetic differentiation exists between these boundaries. Here is an example of Lithia, Tabania, and in east of microplate, there were genetic subdivision in 3 different species: Tabania, Bathymodiolus, and Bathograde crabs. So I applied this IM method in this animal. To do this, first we collected the sample from 20 nodes, 23 nodes to 38 south PL region. So this includes the most extensive distribution area of this species and 10 sampling sites, and we used 12 genetic markers and 4 protein coding gene, 8 non-coding gene, and we first got combined all these markers and generated genetic structure like this. Surprisingly, they are quite match well with the previous known ridge segment northern of the equator and the southern of the equator, and the different region, PAL region. So they are separate roughly, overly separate. However, they share between this boundary, they show some genetic exchange, mixed individuals. So we and also I checked how the genetic diversity, haplotype diversity related with the spreading rate in this area. As the spreading rate increase, we see the decrease of haplotype diversity. This is what is expected from metapopulation dynamics. So the rapid cycle of habitat extinction and rebirth in this region in southern would be the reason for the very low, relatively low haplotype diversity than low spreading area. And finally, we from this previous using this result, we put the data in 3 different populations and then applied 3 population IM analysis, isolation with migration analysis, and they got this result. First, the largest, effectively largest population stay in northern and southern and PAR in that order. And we found the splitting time of PAR and the other ancestor group of 2 population happened around 4 million years ago. It is to me quite surprising because the geologic study said that the formation of eastern microplane happened around 2.5 to 5.4 million years ago. And yeah, I will talk again later. Other different study also independently show very similar result around this boundary. And second separation between NEPR and SCPR happened around roughly 0.8 million years ago. So this is the quantitative estimation of the starting point of this barrier to the evolution of this Alvinella pompejana. And also we found, although it is low, but statistically significant gene flow between these two areas, like here, two arrow indicates real, the highly probable migration or gene flow. And also between these two long time ago separated group, also still has low level of migration. I think this explains some intermixed individual in like here we see intermixed intermixed individual between 2 or 3 areas. So conclusion: genetic diversity of the Pompeii declined toward the super fast spreading portion along the ridge axis, perhaps due to frequent birth and death of vent habitat in that portion, which agreed with the expected from meta population. And unique life history of Alvinella pompejana worms as a pioneering colonizer. Larval development contribute to its high distal capability within each ridge segment, and the dispersal as low as 2 nm and below than 1. Although it is small, but still maintains some genetic continuity across the two boundaries. IM analysis pointed the splitting time at 0.7 or 0.2 millennia across two boundaries. So this is what we got from the IM analysis regarding to the deep sea dispersal barriers. So now on, I focus on two systems, two organisms, Pacific Bathymodiolus and Indian Bathymodiolus medicine recluse and their symbionts. First, Bathymodiolus reside on depends on their nutrition energy on the endosymbiont. Here, this cartoon shows. This figure shows Bathymodiolus azorecus gill tissue and their bacteriocyte internal diagram. The left green is sulfoxidizing bacteria, theotroph, and red one is methanoxidizing methanotroph bacteria. In mid-Atlantic region, this mussel contains dual symbionts, but in our study area, in Eastern Pacific, it contains only sulfoxidizing bacteria. So their symbionts are different from place to place, and it is suggested that these bacteria acquired by host mussels while they are developed from egg to adult from the environment. This indicate this represent horizontal transmission. However, another mollusk, deep sea mollusk in Barents Sea, it is known as vertical transmission mode regarding to about the bacterial origin. So endosymbiont reside in egg cells, so they inherit from parent to offspring like mitochondria or nucleogenes. So this is a typical case of vertical transmission. And also we can think about leaky vertical transmission. Sometimes it mostly come from environment, but still some portion of original symbionts come with the egg cell. So anyway, these deep sea mussel, their symbiont topology and host topology show the very unmatching different topological shape. So that indicates that symbiotic bacteria and host deep sea mussel evolve differently. So they independently evolve. So this strongly indicates the horizontal transmission is the major mode of the bacterial origin. And in EPR, previous studies showed a barrier in eastern microplate, as this pie diagram show different color, different sequence. And another additional study by Johnson et al., they applied more DNA sequence data, and then they found as similar as the previous study, but she also found a very interesting point that in 23rd South area, it contains a mixed individual between mostly blue type and the red type. So they are now recognized as different separate species. So parental species, although two different parent species, only at this side we see some here, or some other place we see some mixed individual resulting from the hybridize between these two parent species. So from this study, I'd like to note, I'd like to say the independent other study, Lorion et al. In 2013, they published the phylogeny and divergence time of deep sea mussel. And then they are based on their time calibration was based on 3 different fossil calibration points. And they calculate the separation between northern and southern deep sea mussel happened around 2.7 million years ago, including the interval is around 1.7 to 4 million years ago. So again, different method based on different evidence, calibration points is quite well matched with the geologic result of the age of formation of eastern microplate, around 2.5 to 5.4. So that is quite interesting point to tell or remember. And for the study of symbiotic bacteria, we had to go a long way from the from getting genetic marker from bacteria. So we sequenced the bacterial genome from the Eastern Pacific Bathymodiolus symbiont. So we are now finishing to reporting this gene with Meva Perez. She is now in the seminar. And so from this genome data set, we selected and we designed the protein coding gene area and the 116 as RNA sequence. And then we did PCR, amplicon, and NGS sequencing and data processing for the population study of bacteria. So the result is like this. The symbiotic bacteria of 2 parental species are also quite well separated. This bacterial gene, protein coding gene, very clearly show this bifurcating or 2 allopathic pattern of haplotypes between northern species and southern species. Southern means PAR. And also when we are interested in one point that how they acquire their bacteria. In 20AS, where hybrid individual exists, regardless of what parental genotype they have, their symbiotic bacteria is northern type at 20AS. So again, the symbionts are acquired from the local environment where the free living stage bacteria exists. This indicates that way. And from our study, we could see more like the first different hierarchical level of differentiation in bacteria. First, clear one thing. From our observation, we made a model for the geographic distribution of symbiotic bacteria like this picture. So if there is a clear physical barrier like eastern microplate, the bacteria in symbiotic bacteria also divided as like here bottom color indicates different genotypes of bacteria. And while their hosts develop, they acquire these bacteria from their local corresponding local areas like this. So and the lizard, purple, green, red only exists in that area, and their host individual contains only this type. And also the different areas where different bacteria exist, they acquire their bacteria symbiont from environment, and then proliferate. These bacteria proliferate within their host tissue. And one more point is the two hosts in the same local, they might show some difference in the proportion of different color strain. So I think this we interpret this as when this environmental fusion happened in the early development of mussel larvae or mussel some certain size of mussel in gill tissue. They start to by chance they amplify their composition, and might be, and in other time they might end up with a different proportion. So there is a chance effect. So from the study of Eastern Pacific deep sea mussel and their bacteria, we found the strong genetic disconnection in both host mussel and their symbiotic bacteria due to the formation of the eastern microplate. The age of the eastern microplate, this much, enabled us to estimate DNA substitution rate of the protein coding gene of bacteria. Like it is calculated as 0.77 to 1.62% per nucleotide per million years. Surprisingly or happily, I found this rate is very similar to E. coli and then the symbionts of aphids and aphids. So not bad to me. Therefore, this information of DNA substitution rate of bacteria will be very helpful for the molecular evolution study of other symbionts in the future, I think. And we ourselves utilize this estimate for the study of the Indian Ocean. So in Korea, the deep sea research project was initiated around 27, and we first we went to cruise 27, and second year we found the new vented field in Indian Ocean Central Ridge area, where we found this only vent site. It has a diffuse type of vent, and here Conca snail and Bathymodiolus mussel, and the other one Gigantidus new species we reported. And here we study with these questions that we want to compare the diverging process symbiotic bacteria in Indian Ocean to that of the Eastern Pacific. So here the green dot indicates new vent field in Indian Ocean, loss of solitaire that is found previously, and this three areas shows different reach segment, and it shows offset of the ridge axis. It's not linear; they are offset, and I think the vent the other vent area, vent field were not observed yet. So from this new finding and new collection of vent organisms, Chimera, they found that OVF on the vent field is their biological communities are much different from previous these triple junction area and solitaire. So From here, we studied the deep sea mussel, Bathymodiolus medicindicus, and collected sample. Some are provided by my former advisor, Bob Hook, and we got this host sample and 3 different DNA loci data look like. So as you can see here, mitochondrial DNA, some areas, some haplotype has green and yellow, green and red here, green and red. But in EF1 alpha gene doesn't show such a pattern. It's just the starbursting haplotype pattern. And collagenase gene here, we could find such a pattern. Here, mostly the red and green, they show more than the blue or yellow. The reason why I'm telling this thing is related with the final our IM result. And again, symbiotic bacteria sample collected from this area, and then we applied homologous gene that designed and found from the Eastern Pacific area, ridge area, and then we applied these 6 different homologous gene of bacteria, and we did PCR amplicon NGS sequencing, and we got this much Illumina leads. So this is kind of many DNA sequence, and then we spread out their sequence according to their unique sequence type, haplotype. As we see here, the green and red are more clustered than blue and yellow. Here, such a pattern exists. And this might be related with the final our result. First, we compare the genetic distance between vent site and bacteria. We found very interesting 3 patterns here. This x axis represents geographic distance between vent site, and the y-axis means mean genetic distance between bacterial gene. So here in EPR, the EPR and PR within their each biogeographical areas, the genetic distance has nothing to do with geographic distance. However, in this EPR, when we compared the DNA sequence between different ridge samples, one from EPR and the other one from PR, their difference was very high. However, here again, we see no relationship between geographic distance and genetic distance. And so this blue area means within this comparison, and their data here, that is blue. And the red means this area and that area comparison, and the result here shown here. And in Indian Ocean, this purple well, comparison of these 4 band field sample comparison. So we see clear increasing relation, the positive relationship between the XY and geographic distance. So we found here ocean-based the leach difference in terms of bacterial diverging process. And we applied the IM model to this bacterial sequence data set from different area, and we pulled their NGS data and randomly selected 100 different leads, and then we applied this data set to the IM program, having three population frame, and we calculate each model parameters like population size, bidirectional gene flow, and splitting time 1 and 0. And before this, we first tried how much each different tree topology or the population topology explain account accounted by our data set. Among these three possible topology, we finally found the most explainable, probable topology of symbiote and host mussel. To me, surprisingly, symbiote bacteria and host mussel show the same topology, although their depths are different. And first, bacteria, we used our mutation right from found from EPR and apply this estimation, and then we finally got this time estimation, like 900 thousand years ago, 700,000 years ago, this split time, and then very statistically significant gene flow direction, bidirection between only solitaire and one-way direction from chiral to solitaire. In case host mussel, they have same topology, but the time depths of splitting times are different from bacteria. That indicates, I think, they evolved independently, but they are governed, dependent on the common geological environment in this Central Indian Ridge. So I think we interpret this data that common environmental geologic the factors influence on both symbiote and host mussel, but due to the different biological characteristics of host and symbiote, the evolution are different in terms of depth and the gene flow. And here we used mitochondrial gene mutation lab from other studies and also other studies. So hosts have different mutational calibration point. So here we are almost ended. This is predicted all in 2002 by Vandover. She predicted the different ridge structure or geologic characteristic would be would influence on the frequency of hydrothermal activity band, and then such a configuration like fast spring ridge, very densely distributed, or here a slow spring ridge where bands are separate away by transform fault or absence of band activity might have different result regarding to the band organism evolution. So we see the according to this Blue and Baker rule, they calculate the fast spring lake has the vent gap distance 3 to 25 kilometers. However, slow to ultra slow spring lake would result in very long distance of gap area between between vents. So, and also in this study, we could find the geophysical components or the effects of geophysical component, the degree of divergence of mussel symbiotic bacteria. The larger is ocean basin, Indian and Pacific. And second, the largest impact is physical barriers such as microplate, is a microplate between these. And third, influencer will be the band field frequency associated with the spring lake of the ridge, as shown in here, the picture. And the physical barrier related to geology effect. So this one is, I think, it's a good calibration point for future study, for the study. So finally, I'd like to conclude that from this presentation, the seafloor geophysical features such as the frequency and distribution of the band activity, transform fault, microplate, and associated deep water current regime along the mid-ocean ridge axis impede the band organisms dispersal, which may account for the general high endemism of band organisms. And the degree of differentiation of mussel symbiont population in the same ocean was highest among InterRidge site EPR between EPR. Okay. And on a scale of intra-ridge, the spreading rate of mid-ocean ridge system, a proxy of the frequency of venting activity, were inversely related to the divergence of symbiotic bacterial vent mussel. Finally, isolation with the migration genetic model helped us to understand the genetic consequence of the biotic and abiotic factors for the dispersal of vent organism and evolution of these organisms. So I'd like to thank my collaborator and my students, graduate students here, Jiyoung and Suyeon, Eunji, Wongyung, and even Meva Perez. Now we are working together in distance area, in distance. And postdoc, Sukjin Jang and Hong Tao Ho and Aji Kumar Patra. And my collaborator for this study, Yong Chung, who developed the MIST program for the over 1,000 genetic IM model. And the Dong-Sung Kim, who is our principal investigator for the Indian Ocean research, he is in Kyoto. And my former advisor, Robert Bryan, who gave me Indian Ocean specimens, and Shannon Jones for the study of EPR mussel, and Corey Slovian for the bacterial study. So thank you for your listening.